Iris tenuifolia. Iris tenuifolia is a beardless iris in the genus Iris, in the subgenus Limniris, and in the series Tenuifoliae of the genus. It is a rhizomatous herbaceous perennial from a wide region over Central Asia, including Afghanistan, Pakistan, the former Soviet Union, republics of Kazakhstan, Uzbekistan and Mongolia, and in China. It has long grayish-green leaves, short stem and pale violet, lilac, pale blue or purple flowers. Description: Iris tenuifolia is very similar in form to the Mediterranean Iris unguicularis, as they both have very small stems and the seed capsules are often hidden within the leaves of the plant. It has a dark brown, thin, short, knobbly tuft, wood-like rhizomes. Underneath, it has a network of fibrous roots. On top of the rhizome, at the base of the leaves, are the brown or red-brown fibrous remains of the previous season's leaves, which act as sheaths for the new leaves. The sheaths can be up to 620 cm to 8 in long. It can be either a single plant or can grow into thick clumps of plants. It has grayish-green, linear, twisted leaves that can grow between 20-60 cm 824 in long and 1.52 mm wide. They do not have a midvein but parallel veins and are acuminate ending in a point. They continue to grow after blooming and can end up as a mass of twisted leaves. The leaves are longer than the flowering stems. It has a very short flowering stem or scape, 10-30 cm 412 in long, although sometimes the stems do not emerge above ground. It has 2 to 4 pointed acuminate, membranous, green between 5-10 cm to 4 in long and 8-10 mm wide, spathes leaves of the flower bud. The stems hold normally 1-3 terminal top of stem flowers blooming in spring, between April and May or late as early June in Russia. The scented flowers are 4-7 cm to 3 in diameter and come in shades of pale violet, lilac, pale blue, or purple. It has 2 pairs of petals, 3 large sepals outer petals, known as the falls, and 3 inner, smaller petals or tepals, known as the standards. The falls are spatulate spoon-shaped, or obovate lanceolate, 4.56 cm 2 in long and 1.5 cm 1 in wide. They have a thin central yellow crest or midvein, dark veins on a pale color, and a band of papilloes or small hairs. The narrower oblanceolate, erect standards are 5 cm 2 in long and 5.9 mm wide. It has a long, slender thread-like perianth tube, 4.58 cm to 3 in long. It has 3 single-colored, style branches, 4 cm 2 in long and 4.5 mm wide. They attenuate narrow slightly and at the tips are toothed. It has a slender 3-4 mm long pedicel, 3 cm 1 in long stamens, and a cylindric 7-12 cm 3.5 in long and 2 mm wide ovary. After the iris has flowered, between late July and early August in Russia, or between August and September in China, it produces an ovoid or subglobose, 3.24.5 cm 1-2 in long and 1.2-1.8 cm 0-1 in wide seed capsule. It has short beak-like appendage on the top. The seeds are oval or turbinate-like, a top-shaped, wrinkled and black-brown to brown. Often, the seed capsule is hidden by the long leaves. Biochemistry. In February 1997, a study was published in which 6 new flavanols, isolated from the rhizomes of Iris tenuifolia, using high-resolution mass spectrometry. In 2005, it was noted that the rhizomes of Iris tenuifolia are the source of the largest number of new 2O-substituted simple flavanols within a single species. Between 2007 and 2011, a study was carried out on chemical constituents and pharmacological activities of Iris tenuifolia and Iris halophila, using chromatographic and spectroscopic techniques. The irises have been used in various traditional herbal remedies, such as traditional Mongolian herb medicine and Uyghur herb medicine. In 2008, Several chemical compounds were extracted from Iris tenuifolia. These included isalpinin, alpinon, arborinon, irilin B, irisone A, irisone B, beta-vulgarin, beta-sitosterol 57, dihydroxyminase 2, 6-dimethoxyisoflavone, 2,5-ditroxyminase 67 methylnedioxyflavanone, irisoida and ethyl, beta-D-glucopyranoside. Also, two new compounds were found, tenuifodione and tenuifone, all found using spectroscopic methods. In 2011, two flavins and a flavonon were extracted from the rhizomes of Iris tenuifolia. Genetics. In 2012, a genetic study was carried out on Iris levigata and several of its closely related Iris species, including Iris insata, Iris setosa, Iris halophylla, Iris scariosa, Iris cottonidea, Iris tenuifolia, Iris blaudoea, and Iris sanguinea. In 2014, the characteristics of phenotypic plasticity and ecological adaptation of Iris tenuifolia from various habitats in Xinjiang, China, were studied. As most irises are diploid, having two sets of chromosomes. This can be used to identify hybrids and classification of groupings. It has a chromosome count, 2 equals 14. Taxonomy. It is written as Zai Yuan Wei in Chinese script and known as Roman 11 Yuan Wei in Pinyin Chinese. The Latin specific epithet tenuifolia comes from the amalgamation of two Latin words tenuis meaning fine or thin and folia mean leaf. It has the common names of Egeria iris. Note, Egeria means water buffaloes or cows pulling in China. Another common name is narrow-leafed iris, or fine-leaved iris, or slender-leaf iris, or silk-leaves iris. It was published and described by Peter Simon Pallas in Rise Russ. Reich. Vol. 3 on page 714 in 1776. It was introduced to Russia in 1812 and was noted as growing in the front garden of Mr. A. Razumovsky near Moscow. It was later published with an illustration in the Gardener's Chronicle 3rd. Series Vol. 59 on page 196 on 8 April 1916. It was verified by United States Department of Agriculture Agricultural Research Service on 2 October 2014. Then, as of January 2015, it is listed as a tentatively accepted name by the RHS. Distribution and habitat. Iris tenuifolia is native to a wide region of various temperate areas of Central Asia, which extends from the Volga through Turkestan into Mongolia and China. Range. It is found in the Western Asian countries of Afghanistan and Pakistan. Also in the Middle Asian countries of the former Soviet Union republics of Kazakhstan, Uzbekistan and Mongolia. Also found in regions of Russia, including Agin-Buryat Okrug, Bashkortostan, Chelyabinsk, Chita, and Siberia. It is listed with Iris blaudoea, Iris humilis, Iris ruthenica, Iris sibirica, and Iris tigridea as being found in the Altai-Saiyan region where Russia, China, Mongolia, and Kazakhstan come together. It is found within central China in the provinces of Gansu, Hebei, Heilongjiang, Jilin, Liaoning, Mongol, Ningxia, Qinghai, Shandong, Shaanxi, Xinjiang, and Zhejiang. One reference mentions Turkey, another reference mentions Iran. Since most others do not mention these countries, they are not regarded as valid. Habitat. It is grown in semi-desert, desert or mild mountainous areas. On sandy steppes on dunes beside sandy riverine grasslands or river banks, on dry coastal sand regions, on gravelly desert-like slopes and in the crevices of rocks. It is also grown at altitudes of 1,000 to 4,200 m above sea level. In northeast China, it is found growing on poor soils on open tree-less plains. Conservation. In 2003, it was listed as an endemic vascular species of the temperate steppe region of Inner Mongolia, China, along with Stipa grandis, Artemisia frigida, Festuca ovina, Thymus serpulum, Caragana microphylla, Coleria cristata, and others. It is included in the Elkin Red Data Book of the Cheetah Oblast of 2002, listed as rared, as rared, as rared, as rared, as rared, as rare. It is now protected in Doria and Copia reserves. Cultivation. Iris tenuifolia is rare in cultivation in the UK. It is rare in cultivation in the US as well. They are more grown by specialized collectors or for scientific and research purposes. It was sometimes used as annual plant and only planted during the summer in the UK in 1800s. It is hardy if sited in a northern continental climate, similar to Nebraska, North Dakota, or South Dakota. It is hardy in parts of Russia. It has been grown in Moscow, St. Petersburg, and Chita. It prefers sandy or sandy loam soils, similar to the desert habitat. It prefers alkaline soils. They prefer positions in full sun. It needs to be kept dry during winter, needing the protection of bulb frames in the UK. It only needs water during the growing period. The plant loses its foliage during the winter, as it is removed by the forces of wind and snow and other bad weather conditions. It then regrows leaves in April and May. It has high drought and heat tolerance desert-like conditions. Propagation. The seed of Iris tenuifolia is rarely used by Western horticulturists, as the plants rarely flower. William Katzendyke notes that it made no satisfactory growth and never flowered. In other flowering regions, the seed can be harvested in autumn washed, fresh or dried. Hybrids and cultivars. Due to its high drought and heat resistance, it could be useful in breeding purposes. Uses. In 2001, a study was carried to monitor the effects of the iris within a herbal remedy for kidney protection was carried out. The iris genus has been used as a traditional folk medicine used to treat a variety of diseases, such as cancer, inflammation, bacterial and viral infections. It was found that compounds isolated from Iris germanica have anti-tumor, antioxidation, anti-malarial parasite, and anti-TB and other positive effects. The roots, seeds, and flowers of the iris are used as ingredients in herbal medicines. They have been used as tocolysis, also called anti-contraction medications or labor represents, and to treat fetal metrorrhagia. On 24 December 2009, a patent was granted for the extraction of Iris tenuifolia, due to its chemical compounds being used in the treatment of Alzheimer's disease.